Hello, my name is Rob Durham,
And I'm John Chappell.
and you're listening to another edition of the Gyrolab Spin. Today on the podcast, we're going to talk about determining binding affinity and kinetics using the solution-based ligand binding assays on the Gyrolab system.
Yeah, really going to highlight how the Gyrolab platform can be used to calculate KD and used for affinity determination. So I think it's a very versatile functionality, and we're finding it can be very useful for our customers to have this option to use this part of the software.
Yeah, I think one of the things that we'll hopefully highlight today is to show how the Gyros is a relatively higher throughput option to screen for affinities, and it could be used as an orthogonal method to other techniques. So let's dive into it. So if we start with thinking about what exactly affinity is, really it describes the overall binding strength of an interaction. And often we think about KD, or the equilibrium dissociation constant, is a measure of how tightly two molecules interact with one another. I think one of the things to keep in mind from a bioanalytical perspective is we're thinking in moles, molar units here, and for folks that have been working in the bioanalytical space, this is different, right, John?
Yeah, I think that's the main thing when setting up the experiments. You need to move away from mass units into molarity, and I know from my own personal experience and working with people that can cause some difficulty. So I think we will focus on a future podcast on very much more on how to set the experiments up with practical examples. But today really is an overview of kind of the theoretical basis of affinity and how we can use the Gyrolab to do affinity calculations.
Yeah, and so just to very simplistically think about this in terms of these kinetic rate constants. So we have two interactants, A and B, and they're going to bind to one another, and that can be described with a kinetic parameter, which is the on rate and the off rate. And KD really is a ratio of the off to the on rate. And I mean, this is the thing that can make it confusing, is that ratio is inverse of what you think. So a low number means high affinity, and a small number means
No, exactly,
low affinity,
exactly.
Right, high affinity, right? Yeah. So let's dive into that a little bit more detail. So we're thinking about high affinity here. That's really describing a tight binding affinity between the interactants. And you can see here in this figure, big AB complex means that at equilibrium, we're going to have a relatively high concentration of our interactant, of our complex, and relatively low of the interactants. And so again, this high affinity then really gives us a number from a KD perspective that's small, and picomolar to femtomolar.
Yeah, I think the important thing to consider is the higher the affinity of an interaction, the longer it actually takes to reach an equilibrium. So high affinity interactions means you actually have to leave the reaction longer. Yeah, which is conceptually the wrong way around for me.
So. Yeah, it's a bit counterintuitive, but again, it's all about that off rate, right? So you have to let things settle, and I think about it like things settling, coming to some sort of an equilibrium, and you just have to wait for that off rate to occur before we establish that real equilibrium. So conversely, here we have then low affinity. Again, weak binding between the interactants means you're going to have a relatively higher concentration in the unbound state. And again, as we talked about, it's counterintuitive, but you achieve equilibrium much more quickly.
Yeah. So it's actually much easier to set up experiments with low affinity. You don't have to leave them as long to reach equilibrium.
Exactly.
So affinity really is a fundamental biophysical attribute, and particularly when it comes to drug development, the affinity of your drug to target interaction is an important part of discovery. Mm-hmm. So it's a way when you're actually trying to select potential drug molecules, you're going to look at the affinity of binding, and maybe you will select molecules with high affinity compared to low affinity, depending on what you're trying to achieve. So it's a good way of ranking molecules. So pretty much affinity is used throughout drug development, right from early research all the way into, when you're manufacturing a drug. It becomes a critical attribute of that molecule to assess the affinity of binding to its target.
Right, and so as longtime immunoassay developers, another key quality attribute for us is high affinity reagents for selecting reagents to build really good assays, right? So
yeah.
Affinity is going to be one of those key parameters that's going to determine the sensitivity of your capture reagent.
Yeah. And also when you look at assays like the Gyrolab, where you need to label reagents to actually incorporate them into your assay, the labeling itself can potentially affect the binding affinity. So I know people want to use affinity measurements to characterize reagents when they make them, and also to allow for like batch comparison, because it can be a part of how you monitor the performance of your reagents over time.
Yeah, so, and for assays that may go on for years and years during the development of a drug or the post-marketing surveillance of a drug, you're going to have life cycle management and being able to manage the batch-to-batch variability of your key critical reagents is going to be very important.
No, exactly.
Well, when looking at the affinity landscape, there's certainly no shortage of options for someone to dive into, make measurements of affinity. And of course, probably the gold standard for everybody is BioCore with the solid phase measurement, and then there's others, the FortiBio and Cartera, which also uses solid phase technique. The Gyros, along with a number of other options, can be used for in-solution measurement. In-solution is really a classical way of doing affinity, and classically would be done with an ELISA assay in a plate. Again, in solution gives you the option of measuring things without having to necessarily label your interactants.
So there are some limitations with current technologies. As mentioned, the gold standard is SPR. This can be inaccurate, particularly when there's a really high affinity, when there's a very slow off rate, which can also lead to very long instrument run times. And effectively, in a surface-based technique, you're actually immobilizing one interactant. So that may not be reflective of the biological situation, or it may be for something like a cell surface marker, for example, but obviously you need to think of what the interaction actually is. But it is the gold standard, and you'll see when you look at the literature and you trying to look at a binding affinity, most of the time the number quoted will probably be from SPR. Right. But obviously we're here to talk about in-solution assays, which is an option on the Gyrolab. This is very much a flow-based technique. A lot of the technologies that do in solution use quite large sample volumes, because you're doing a lot of dilution of samples. But that using Gyrolab, we do use less sample volume than other techniques, which is good. If the reaction is unstable, for example, if you've got really high affinity and you need to leave for a couple of days, you have to ensure that your reagents are actually stable for that time. So if there is any instability in the reagents, then in solution may be a problem. Also in classical ELISA, for example, you're incubating your interactants in your assay. So if there's any equilibrium shift as part of the assay step, this can affect you can get an overestimation of the affinity, etc.
Yeah, that equilibrium shift is one of the advantages on the Gyrolab. And then I think the other thing too, or the lack of equilibrium shift rather on the Gyrolab is an advantage. And the other thing is the parallel processing on a CD means that you're not doing serial sampling, and you don't have the delays in time and larger sample volume that's required for some of those other in-solution techniques. So let's take a look at in-solution affinity determination on the Gyrolab.
So the important thing within solution determinations is that high affinities can be determined, and actually the interactants are not modified, and the interaction is performed in solution, so it does mimic the biological system. As close to the true affinity constant can be obtained. And the affinity of the ligands binding to receptors on cells and also can be determined. So that's an area I know that many customers are working on, like cell-based affinity determination. And as Rob's mentioned, because of the microfluidics, this does minimize the equilibrium shifts because the incubation time on the Gyrolab are in the seconds rather than in the hours as part of the assay steps.
So here you can see that we're really setting up a standard curve that's very much like a PK assay, where you have the capture reagent is binding to the target antibody or the drug, the therapeutic drug, and capturing it out of a solution, and then we're coming back with an appropriate detection molecule to measure the amount that's bound to the column. So in this standard curve, you can see a response versus concentration, and grams per liter is the units there that we're thinking about. Now, when we make the shift to affinity, we're going to be thinking about molar concentration of our fixed interactant. So we're going to pick one concentration of our drug and then compete that with increasing concentrations of the target. And both the parameters are going to be measured in moles so that we can understand the stoichiometry of the binding interaction. And so here then you can see that we've selected one point on our standard curve to the left, and we pick something that's around 10-fold over background so that we have enough of a signal that's measurable, and then we can measure a decrease when we compete that signal away using increasing concentrations of our molar concentration of our variable interactant. The point here is that we're using the same format with both assays. It's just the setup of the assay is different.
Yeah, I mean, you have to start by obviously pre-incubating your drug and target in solution, and then what hasn't reacted will then bind it within the assay. So you start off, when you actually design the experiments, you start off by doing a concentration curve as on the left, and then based, as Rob said, based on the signal to noise, you select a single concentration of your drug, and then you vary the concentration of the target, and that causes inhibition of signal. And actually the selection of concentrations is actually quite an interesting thing, and actually where a lot of the complexity in setting the experiments up are. And actually we will do another podcast series where we're really going to focus a lot more on the experimental setup when we go through how you customers and people working on system can actually design these affinity experiments.
Yeah, I think that's important to really go beyond the theory and get into the practical aspects of that. We'll definitely tackle that on the next one.
I think
part two.
Exactly. I think affinity conceptually can be quite complex for someone, particularly that's what used to do in normal bioanalytical assays. But when we break it down, I think it's something that any scientist can achieve in their lab.
So this graphic just shows how you would sort of simply set up the solution affinity experiment. So you're going to prepare your affinity samples by mixing a fixed concentration of your one interactant with a variable concentration of the binding partner. And this will form a competition affinity series in the microplate. Then we can let that mixture incubate until it comes to some sort of an equilibrium. We can try various different times. And
I think that's the thing, is you don't always know what the interaction affinity actually is. So you may want to leave it for a number of, maybe want to do the incubations for 2 or 3 hours, and then maybe do an overnight incubation, then compare the data and see, to give you, and see which gives you the most accurate determination of the KD. But the good thing about the Gyrolab being so quick, you can set up all these experiments and just run them, and you get the results in an hour.
Yeah, one right after another.
Exactly.
So one of the nice things about the Gyrolab software is it allows you to relatively, in a straightforward way, set up a series of experiments to measure affinity. So in this plot, what we're looking at is a normalized response of free versus a molar concentration of the variable interactant. And you can see that at low concentrations of our fixed interactant, we're in a situation where the we're KD controlled. That is, where the concentration of our fixed interactant is at or below the KD. And that leaves us with all of the interactant being consumed before we've sort of consumed all of the variable interactant. Versus in a stoichiometrically controlled situation, we can measure more accurately the fixed concentration, the active concentration of the target. And here, the fixed concentration is several orders, 10 times KD, in an effort to sort of consume all of the variable interactant as we approach zero.
Yeah, I think very much in the next podcast where we go through the experimental setup, we'll go much more into detail how to define a KD control curve and a stoichiometric control curve.
Yeah.
Because it's very important, actually, part of the experimental setup.
Right. So in this 3D plot, then, we can see how varying the interactions, the fixed interactant in various different experiments, moves us into the Z direction in this three-dimensional plot, so that we aim to get at that place in the middle, a purple curve, where we have a sweet spot where we have a accurate KD determination as well as a accurate fixed active concentration determination. The problem is, in reality, you don't know these things parameters up front, and so you're going to have to do several different fixed concentrations in an effort to sort of understand which setup is giving you the most accurate KD and the most accurate.
Yeah, so it's trial and error.
Yeah,
running various curves to see.
A bit, but I think one of the things that we'll show in a minute is, the nice thing about the software is it allows you to sort of combine multiple curves together so that you can do the experiment in one go and then have a pretty good setup to determine what your KD and your active concentration is. So again, this multi-curve analysis with the latest version of the Gyrolab Affinity software module allows you to do combine multiple curves to allow you to share the KD and the F-tote for a group of affinity curves, and that allows you to make sure you've got good confidence in determining both parameters.
Yeah, it proves your calculation, your KD. It basically pulls the parameters together and it makes it tighter and more accurate in determination. And it's really good because when you're setting up affinity, you're going to run different fixed concentrations. So it allows you to combine these to give you a single KD determination.
And I think what you're doing here is sort of setting up an assay. And once you've identified what those parameters are, to have a fixed concentration for a KD controlled curve and what's the optimum fixed concentration for a stoichiometrically controlled curve, then you can use those two parameters to run a screening study. We'll talk a bit about, show how you do a screening assay once you've identified what the optimum parameters are. But getting there is the first step, and that's what this multi-curve analysis allows you to do. So let's take a look at an example of affinity determination on the Gyrolab.
So for this practical example on the Gyrolab, we're actually going to look at some TNF-alpha antagonists. So TNF-alpha antagonists are a big class of treatments for inflammatory conditions. So we have a number of products, including things like infliximab, adalimumab, Enbrel, and also second generation. So we have biosimilars starting to come through, and also second generation such as golimumab. It's a big class. Inflammatory conditions are very common, so it's a big industry, and things like rheumatoid arthritis, through to psoriasis and ulcerative colitis, Crohn's disease. These molecules are used for and very effective treatments for these conditions.
So once we've established the assays here, you have a TNF-alpha dilution series where we're going to cover a pretty broad range of the variable interactant, between 20 nanomolar down to 2 picomolar. And then the TNF-alpha antagonist is the fixed interactant with the binding concentration for 10 picomolar for the KD controlled curve and around 500 picomolar for the stoichiometrically controlled curve. So once you have those parameters identified, now we can set this up as a screening assay where we can look at all the different various TNF-alpha antagonists that we want to screen. And again, we're measuring the free drug, the TNF-alpha antagonist in this case, by setting up our competition curve. And then once it's come to equilibrium, then it's a simple matter of running the samples on the Gyrolab bioAFFINITY and measuring free drug.
Yeah, I mean, the affinity is going to be very high. So as you can see here, the reaction is being left for 72 hours, so 3 days. So, which is obviously a long time, but it reflects it being high affinity. But after the experiment is set up, this experiment is left to sit. And then, when you actually come to read it and put it on the instrument, it'll take an hour to generate the results.
Yeah, I think probably, and if you got the setup worked out, you can probably set this experiment up in 45 minutes to an hour, let it incubate until it comes to equilibrium, and then it's only going to take an hour to screen your whole set of assays. So from a user standpoint, it's not a lot of active time.
So here we've looked at the data. So this shows the affinity determination of the 5 TNF-alpha antagonists. So we've run all 5 in a single experiment, and then we can then calculate a KD. So as you can see, we've actually calculated KD in the low picomolar, so from 8 to 20 picomolar. All the different antagonists are given very similar affinity. And they're all binding in the picomolar range, and all this can be determined in one Gyrolab affinity run. So it's very good for actually ranking or comparing different molecules that bind to the same target.
Yeah, I think if you have this worked out, you could probably screen. I think we've worked it out. It seemed like you could probably screen 5 to 10 different molecules on one CD if you were judicious about if you really had the conditions worked out.
Yeah, I'm actually interested to know how our calculated picomolar affinity actually reflects what's in the literature. So it's something when I would like to look following this.
Yeah, absolutely. So in the same experiment, we used the stoichiometrically controlled conditions at 500 picomolar to determine the active concentration of the TNF-alpha antagonist. And here we want to confirm that we're getting about the same active concentration across all of the different experiments. And that says we've got good control over the concentration of the drug as well as the variable concentration. And here you can see that we're seeing about the same active concentration, right at normalized to about 100% across the board.
Which is pretty good and very consistent.
Yeah.
When it comes to the time to reach equilibrium, so the time to reach equilibrium will depend on the kinetic binding properties of the interactant. So here we give you a kind of a rough guide of how long you need to leave to actually reach equilibrium. So we're suggesting for around, if you expect the KD to be around 1,000 picomolars, then you need to incubate around 12 hours. But if you're looking at much higher affinity, for example, less than 5 picomolars, then you may need to leave for approximately 3 days. So it's just really a guide for how long to actually leave your reaction. And you can do a time course experiment where you leave your reaction for increased time and then compare the kind of the calculated KD.
Right, and you can see in this example, as you increase the incubation time, that KD value sort of stabilizes and shifts to the left until we've achieved, true equilibrium at the point where we're measuring it. We're going to underestimate it at earlier times.
So hopefully we've given you a good overview of in-solution affinity determination on the Gyrolab. I think the important thing is that we can measure KD values down to low picomolar. It does do solution-based measurements, which does minimize biological system. It's a very useful thing that you can do on the instrument, and even if you're not doing affinity determinations regularly, it's something maybe you can use to characterize reagents or to understand some of the results you're getting. It's a very useful tool. The software is really good. It's very intuitive, and I think it's something that everybody out there can give a go.
Yeah, I think it's again one more tool in your toolbox. Just highlights the nature of the Gyrolab system as being an open platform. It gives you lots of flexibility to use the system for a wide variety of applications.
Yeah.
So thanks to everyone for joining us today on this discussion about affinity. Again, as always, please hit the subscribe button. If you liked what you heard, and always please reach out with your comments, questions, and feedback. We're really interested to hear what you have to say.
Yeah, we're really would like your feedback, any suggestions, comments. Please contact us. I think we find we really want to increase the amount of podcasts that we do, so any suggestions you can give us, because we really think that these hopefully will be useful to you.
Right. And so I think as a follow-on to this one, or at least a couple more that we had ideas, we'll want to get into the practical setup of an assay. We might do some more advanced affinity determinations and have a guest speaker come on and join us. So we're excited about those for the future as well.
Yeah, I mean, it is possible using kind of time course experiments to perform kind of more kinetics, things to calculate the K off and K on, but it's intense mathematics, so we'll leave that to a future podcast.
Perfect. Until next time.
Yeah, take care, everyone.